If you have a brown bottle of hydrogen peroxide in your cabinet, you may be holding one of the most misunderstood products in the garden. Pour it onto potting soil and the surface can erupt in white bubbles. Soak an old seed and it may germinate faster. Apply the wrong concentration to a healthy root, however, and the same bottle can damage the very plant you meant to rescue. That is because hydrogen peroxide is not fertilizer, bottled oxygen, or a magical cure. It is a fast, non-selective oxidizer, and what it does depends on where it lands, how strong it is, and what is living there. Gardeners have used peroxide against fungus gnats, root diseases, dirty pots, algae, contaminated tools, stubborn seeds, and struggling hydroponic roots. Some of those uses are legitimate. Others work only under controlled conditions, and several of the most popular online tricks leave out the one detail that determines whether the plant improves or quietly gets worse. So before another capful goes into a watering can, here are 11 things experienced gardeners understand about hydrogen peroxide, beginning with a reaction almost everyone misreads. Number 11. The bubbles are not proof that peroxide found a disease. The moment hydrogen peroxide reaches soil, dead leaves, damaged tissue, or a dirty pot, it begins breaking down. The familiar formula is H2O2. As it decomposes, it becomes water and oxygen. Enzymes such as catalase, along with metals, microbes, and organic material, can make that reaction happen much faster. The oxygen escapes as the white fizz you can see, but the reaction cannot identify what is present. Healthy plant tissue, harmless soil organisms, compost, and decaying bark can all accelerate the breakdown. Even a cut potato foams when peroxide touches it because living cells contain enzymes that dismantle peroxide. The bubbles reveal a chemical reaction, not what caused it. This is the first detail that changes every trick that follows. Fizz does not prove that root rot is dying. It does not prove that fungus gnat eggs were destroyed. It does not prove the roots needed oxygen. And more foam does not automatically mean better cleaning. It simply shows that peroxide is being consumed, often within moments of contact. Number 10. It can strike fungus gnat larvae, but it cannot end the infestation by itself. Fungus gnats are attracted to damp growing media rich in fungi and decaying organic material. The adults are the tiny dark flies that rise when a pot is moved, but the translucent larvae live below the surface. That hidden stage is why a soil drench is so tempting. Some university extension guidance describes a mild mixture made from 1 part ordinary 3% peroxide and 4 parts water as a way to contact larvae in potting soil. When that diluted solution reaches exposed larvae, its oxidizing action may knock down some of them. The fizz also makes the treatment feel immediate, but peroxide has 2 limits that the bubbling hides. It breaks down rapidly in organic soil, and it acts only where the liquid makes effective contact. Eggs, larvae, tucked into protected pockets, pupae, and adults flying around the room may survive. A few days later, the pot seems to have produced gnats again. The permanent control is less theatrical. Allow the upper layer of mix to dry to the degree the plant can tolerate. Empty standing water. Improve drainage and remove decaying debris. Catch adults with yellow sticky cards so fewer eggs are laid. For a persistent larval population, a BTI product labeled for fungus gnats can target that life stage without pretending one fizzy watering changes the habitat forever. Peroxide can be a contact treatment. Moisture management breaks the breeding cycle. Confusing those 2 jobs is why the flies often return. Number 9. The most impressive seed trick is real, but it is not universal. Seeds do not simply wait for water. Inside them are chemical signals that control dormancy, metabolism, defense, and the moment growth begins. Hydrogen peroxide also exists naturally in plant biology as a reactive oxygen species and signaling molecule. In carefully controlled research, exposing certain seeds to a specific peroxide treatment has helped shift them from dormancy toward germination. USDA researchers working with sugar beet seed reported a protocol that improved germination by as much as 70%. Their genetic analysis suggested that the treatment reduced activity associated with dormancy while promoting activity connected with growth and cell proliferation. But the words certain, specific, and controlled matter. A protocol developed for sugar beet germplasm is not an automatic recipe for tomatoes, peppers, lettuce, orchids, or a packet of expensive native seed. Species differ. Seed coats differ. Pretreatments already applied by the supplier differ. Concentration and exposure time can separate stimulation from injury. Peroxide strong enough or applied long enough to suppress surface organisms may also reduce vigor or harm the embryo. So the useful secret is not to soak every seed in the same glass. It is to look for a tested protocol for that exact species and purpose. If the seed is commercially treated, follow the supplier's directions. A research result can reveal a powerful biological effect without becoming a universal kitchen table formula. Number 8. Peroxide is often more dependable around the plant than inside it. Imagine pruning a diseased stem and then carrying the same shears to a healthy plant. The blade may look clean while microscopic bacteria, fungi, or viruses remain on its surface. Used pots, propagation trays, greenhouse benches, irrigation parts, and plant stakes can also move problems from one crop to the next. This is where peroxide-based products have a serious horticultural role. Extension guidance lists labeled hydrogen peroxide or peroxide and peroxyacetic acid disinfectants for hard, non-porous greenhouse surfaces, pots, trays, tools, benches, and equipment. They are oxidizers, designed to reduce microorganisms when mixed and applied exactly as the label requires. But disinfecting does not mean spraying over a crust of soil and walking away. Organic debris can consume the active ingredient before it reaches the organisms underneath. The surface is cleaned first. Then the correct product is applied at the labeled dilution and kept wet for its stated contact time. Some products require rinsing. Others have restrictions around plants, seeds, food contact surfaces, or particular materials. A pharmacy bottle and a registered horticultural disinfectant are not interchangeable. A formulated product has directions for a defined site, concentration, contact time, and target. If the job is preventing disease from traveling on equipment, the label, not the bubbles, is the operating manual. Number 7. That green film is more than an ugly stain. Algae grows where light and persistent moisture meet, on greenhouse floors, capillary mats, propagation benches, pots, trays, and constantly wet growing media. It creates a slick surface and helps maintain the damp environment favored by fungus-gnats and shoreflies. Certain peroxide-based horticultural products are labeled as algicides or sanitizers on specified surfaces. On an emptied, pre-cleaned tray or bench, they can oxidize algae and associated microbial buildup quickly. This is one reason commercial growers may use peroxide chemistry even when they would never pour household peroxide repeatedly into every planted container. The hidden step is to remove the condition feeding the film, repair leaks, improve drainage, stop leaving trays full of water, increase airflow where appropriate, and clear organic debris. On soil, rethink the watering schedule instead of repeatedly bleaching the green surface. If light and water remain, the algae has everything it needs to rebuild after the oxidizer is gone. And never assume a surface sanitizer is safe to spray across leaves or roots. Some EPA-labeled products used in horticultural settings explicitly require plants to be removed before disinfection. Others are registered for particular plant applications. Similar-looking bottles can have completely different instructions. The site named on the label is part of the treatment. Number 6. Peroxide can make root rot look better before the plant is actually safer. A root system needs oxygen, but healthy potting soil obtains it through pore spaces. When those spaces remain full of water, gas exchange slows. Roots become stressed, fine roots begin to die, and water-loving pathogens may gain an advantage. This is the situation behind the seductive idea that peroxide oxygenates the soil. Yes, decomposition releases oxygen. Briefly, gas bubbles may move through the wet mix, and oxidizing contact may reduce susceptible organisms. A wilting plant may even perk up temporarily. None of this rebuilds the air spaces that collapsed in a compacted, saturated pot. If the drainage hole is blocked, the pot is sitting in water, the mix is broken down, or the roots are already decaying, the physical cause remains after the peroxide disappears. The next heavy watering recreates the same low-oxygen environment. Worse, damaged roots may be exposed to an oxidizer at the moment they are least able to tolerate more stress. Real root rot recovery begins outside the bottle. Confirm the problem rather than diagnosing by a yellow leaf alone. Remove the plant carefully, inspect for brown or water-soaked roots, correct the container and drainage, replace unsuitable medium when necessary, and adjust watering to the plant and environment. Severe disease may require disposal or a product specifically labeled for the diagnosed pathogen. Peroxide can be an ingredient in some registered disease management products, especially in professional production. That does not turn a household soil drench into a cure for every brown root. Temporary oxygen and permanent aeration are not the same thing. Number 5. Hydroponic growers use peroxide for a different reason than soil gardeners think. In deep water and other hydroponic systems, roots depend on oxygen dissolved in the nutrient solution. Air pumps and surface agitation provide continuing gas exchange. If circulation stops or the water becomes warm, oxygen can fall while microbial growth accelerates. Diluted peroxide is sometimes used in hydroponics to raise oxygen briefly and reduce microbial buildup. But notice the difference. A hydroponic reservoir is measurable. A grower can monitor temperature, pH, electrical conductivity, and dissolved oxygen. The system may be intentionally run as a relatively sterile setup rather than a biologically active one. The grower can also calculate the volume of water and use a product intended for that system. Potting soil is not a reservoir with a known chemistry. It contains bark, peat, coir, compost, minerals, roots, fungi, bacteria, and countless reactive surfaces. Peroxide may vanish much faster and distribute unevenly. Copying a hydroponic dose into a flower pot therefore ignores the environment that determined the original practice. Even in hydroponics, peroxide does not replace an air pump. Its oxygen release is temporary, while roots consume oxygen continuously. It also cannot repair a failed pump, overheated solution, clogged channel, light leaking into the reservoir, or dead roots decomposing in the water. The durable fix is to restore circulation, control temperature, block light, clean the system, and remove the source of decay. That difference separates a useful treatment from a costly plant trap. Peroxide is sometimes a management tool in a defined system. It is not a substitute for designing that system correctly. Number 4. Every soil drench hits allies as well as enemies. Healthy growing media is not empty dirt. Around the roots is a crowded biological zone where bacteria and fungi consume organic material, compete with pathogens, cycle nutrients, and interact with the plant. Some are harmful, many are neutral or beneficial. Hydrogen peroxide does not read name tags before it oxidizes what it touches. That non-selective action is exactly why peroxide can be useful as a disinfectant on an empty tray. It is also why repeated soil drenches deserve caution. A concentration capable of injuring a pest or pathogen can also disturb organisms the gardener wanted to keep. In a living soil, compost-rich mix, or inoculated medium, routine peroxide use may work against the reason those biological ingredients were added. The effect will not be identical in every pot because organic matter rapidly consumes peroxide and plants produce enzymes that defend against it. That variability is not proof that any dose is harmless. It means the actual exposure at the root is difficult to predict from the amount poured at the surface. If the goal is to rescue a plant from a confirmed problem, a limited treatment supported by reliable guidance may be reasonable. If the goal is simply to make healthy soil bubble once a week, there is no clear problem being solved. Good drainage, correct moisture, appropriate temperature, and clean growing practices support roots continuously without repeatedly resetting their microbial neighborhood. Number 3. Spraying the leaves can turn a tiny spot into a large burn. Online garden recipes often jump from soil to foliage without acknowledging that leaves are living surfaces built to control water loss and gas exchange. Hydrogen peroxide can damage cell membranes through oxidation. Tender new leaves, thin foliage, seedlings, flowers, and already stressed plants may be especially vulnerable. Sunlight and heat can increase the stress surrounding a spray application, and droplets can collect at edges or inside folded tissue. The first damage may not appear while the leaf is wet. Hours later, pale speckles, browned margins, translucent patches, or collapsed tissue emerge. A gardener may mistake that injury for the disease and apply even more peroxide, expanding the damage in the name of treatment. Some commercial peroxide formulations are labeled for particular crops and plant diseases. Their directions specify concentration, timing, coverage, protective equipment, and restrictions. That is fundamentally different from misting undiluted 3% household peroxide over every leaf, because a social media clip showed powdery mildew disappearing. Before treating foliage, identify whether the white material is actually fungal growth, mineral residue, insects, or natural leaf structure. Remove severely affected material when appropriate. Improve airflow and spacing. Water in a way that keeps foliage from staying wet unnecessarily, and use a product labeled for the crop and disease if chemical control is needed. A spot test can reveal obvious sensitivity, but it does not turn an unlabeled use into a proven one. Waiting long enough to observe the test area matters because oxidative injury is not always immediate. Number 2. One capful is not a concentration. Household hydrogen peroxide is commonly sold at 3%, but gardeners can also encounter stronger beauty products, food processing grades, and concentrated horticultural formulations. A tablespoon of one is not equivalent to a tablespoon of another. Some high-strength peroxide can cause serious burns and should never be handled like the brown bottle from a medicine cabinet. Even 2 bottles marked with peroxide may contain different stabilizers or additional active ingredients. A professional product combining hydrogen peroxide with peroxyacetic acid behaves differently from plain 3% solution. The label may call for gloves, eye protection, ventilation, a precise dilution, and a specific contact time. Moving the concentrate into an unmarked spray bottle removes the information most likely to prevent an accident. Strength also changes after storage. Hydrogen peroxide is packaged in a dark container because light encourages decomposition. Heat, contamination, and contact with reactive material can accelerate it. A bottle left uncapped in a sunny potting shed may gradually become mostly water, while a fresh bottle from another source may be far more active. Never pour used solution back into the original container, and never seal a contaminated mixture in a vessel where oxygen gas could build pressure. This is why recipes measured in capfuls are unreliable. The cap size, starting strength, final volume, plant sensitivity, and purpose may all be unknown. Start with the product label and a recommendation for the exact use. More peroxide is not a faster version of the same treatment. Beyond a point, it becomes a different hazard. Number 1. The most important peroxide trick is knowing what never belongs in the same bottle. A garden shelf may hold vinegar, bleach, ammonia cleaners, copper products, fertilizers, pesticides, soaps, and acids. Combining products because each works separately can create a new chemical exposure with risks that neither label was written to manage. Hydrogen peroxide and vinegar can form peracetic acid, a powerful irritant used in carefully formulated commercial disinfectants, but not something to improvise in a kitchen sprayer. Mixing peroxide with household bleach can drive rapid reactions and gas release. Combining cleaners can also create heat, pressure, irritating vapor, or compounds that damage lungs, eyes, skin, plants, and equipment. The safe rule is simple. Unless the product label specifically directs the combination, do not mix it. That includes the clever idea of adding peroxide to a pesticide tank to make it stronger. The oxidizer may break down the active ingredient, change the pH, injure the crop, or make the mixture unsafe. Apply only label-approved tank mixes and never use a food or drink container to measure or store garden chemicals. The same restraint applies to wound treatment. Pouring peroxide into a freshly cut stem or root may destroy exposed cells needed for recovery. Plants already generate reactive oxygen compounds as part of their own signaling and defense. Flooding injured tissue with more is not automatically helpful. So, what do gardeners really know about the brown bottle? Hydrogen peroxide can disinfect appropriate empty surfaces when the product is labeled and given enough contact time. A mild, supported treatment may contact fungus-gnat larvae, although moisture control must finish the job. Species-specific protocols can influence seed germination. Defined hydroponic systems sometimes use controlled peroxide additions. Registered horticultural formulations can suppress certain algae and plant pathogens, but peroxide cannot diagnose disease by bubbling. It cannot rebuild compacted soil, unblock drainage, provide continuous aeration, distinguish beneficial microbes from harmful ones, or make an unidentified leaf problem disappear safely. The reaction is quick, visible, and satisfying. The condition that caused the problem is usually slower and less dramatic. That is the final secret. The best use of hydrogen peroxide often happens before the plant is in danger, cleaning the right equipment, following a tested protocol, and preventing contaminated tools, trays, or water systems from spreading a problem. Once a plant is failing, the bottle should never replace diagnosis. When the peroxide touches the garden, 3 questions matter more than the fizz. What exact problem has been identified? Is this product labeled or reliably supported for that site and plant? And what physical condition, water, drainage, airflow, sanitation, temperature, or light allowed the problem to develop? Answer those first, and the brown bottle becomes a limited, understandable tool. Ignore them, and 11 clever tricks collapse into one expensive mistake: treating the reaction while the real cause keeps growing underneath it.